Hello dear students, welcome back to PharmWise Academy. In our previous lectures, we have studied about the cell and its organelles. So now in this topic, in this video, we are going to discuss about the various cell junctions. So let's get started. So basically, the cell junctions are basically we have to discuss about 5, but here is the classification. The cell junctions can be divided into two types. The first one is the cell to cell junctions, and cell matrix junctions. Okay, the cell to cell junctions and cell matrix junctions. Now what are you watching these? The tight junctions, adherence junction, desmosomes and gap junctions. These are all the examples of cell to cell junctions, because these junctions are present in between the cells. Okay, in the cell matrix junctions there are two junctions present. The first is hemidesmosome. Hemidesmosomes and another is adhesion plaques. So we are not going to discuss in detail about the adhesion plaques because they are rarely present, but we are going to discuss about the hemidesmosomes. So let's start it from the first, that is tight junction. So what do you mean by tight junction? Here is the representation. Can you see? This is the plasma membrane of one cell and this is the plasma membrane of another cell, and in between these two cells there are some protein complexes present which are binding or which are joining the two cells each another, right? Can you see this? Yes, these are the protein complexes. Now what is the protein complexes? What are their contents? That is the protein molecules, occludin, claudin, etc, etc. These are all the protein compounds which are form protein complex, and they are present transdermally between the two cells, right? Here it is present. What is the structure? It is found in epithelial tissue. Mostly the tight junctions are found in the epithelial tissue, where the lining epithelial tissue mainly provides the lining of the inner surfaces in our body. So if a lining has to be provided, then the cell junctions should be very tightly attached. The cells must be tightly attached to properly define a cell wall, right? So therefore it is found in the epithelial tissue. As the name suggests, tight junction are, in these tight junctions, the cells which are attached, they are held very tightly, right? And next, the transmembrane proteins from adjacent cells interact to seal the intracellular space, right? This is one cell and this is another cell, and in between this space it is called the intracellular space. Now in this intracellular space is completely sealed by these protein molecules. They intersect with each other, right? Now what are its functions? It forms a barrier to prevent leakage of molecules between cells, right? If it is provide lining and it forms a barrier, then it prevents the leakages of molecules, right? Okay, and maintains tissue polarity by separating apical and basolateral surfaces. Now what do you mean by this? Basically, if a cell has to be oriented which is top and which is bottom layer, then by this tight junction it is clearly visible that we can differentiate between the apical cell and basolateral side. Means we can say that this is the bottom, this is the top, and this is the bottom, right? Now what are some examples of tight junctions? Okay, this is the structure and function, but where these tight junctions can be seen, right? I will give you examples. The first example is the tight junctions are present in the collecting duct of nephrons, right? What is nephrons? Nephrons are the structural and functional unit of kidneys. They are present in kidneys. In nephrons there is a structure called collecting duct, and in that collecting duct the lining is provided by the tight junctions, right? And it is also present in bile duct. It is also present in the bile duct which is coming out of the liver, okay? And it is also present in blood-brain barrier, right? Have you studied about it earlier? Okay, no problem. You are going to study, however, in further classes. And also the blood cerebrospinal fluid barrier, blood CSF barrier, and BBB, that is blood-brain barrier. In these cell linings, you can see the tight junctions, right? Next, the next second one is the adherence junction. Now, what do you mean by adherence junction? Adherence junction means one cell is held by another, but the difference is here the protein molecules, here protein complex is absent, but rather many protein simple molecules are present, which are the cadherins and catenins. Okay, these are the important words we have to note down. The cadherins are present extracellularly, means there are two proteins, cadherins and catenins. Catenins are present in extracellular matrix, where catenins are present in intracellular matrix. Here you can see catenins, and here is the cadherins. The cadherins are projected outside of the cell, and the cadherins are attached to another cadherin of another cell, of the adjacent cell. Okay, by this the one cell is attached to the another cell. Here you can see the binding, and inside the catenins are linked to the actin filaments inside the cell. Okay, actin filaments which are present inside the cell, they are directly linked to it, right? Catenins are you can see these greenish molecules, these are catenin molecules. These catenin molecules are again directly linked with the actin filaments. Now what happens means by the help of these, what is the main purpose of cadherins and catenins? It is because when for example, in the case of cardiac muscle cell, okay, in the case of cardiac muscle cells, due to the contraction and relaxation of the heart, there will be tightening and continuously tightening and loosening of the cells takes place. So in that case, to hold the cells tightly, the actin filaments, actin filaments are involved in the movement of the fibers. Actin and filaments are involved in the movement of fibers and muscle tissues. Therefore, when actin moves, along with that catenin molecules are also moved, and they ensure that the cells are not driven away by each other. So they help in keeping the cells intact, right? Cadherin protects protein connects cells. Cadherin links to actin cytoskeleton inside the cell, right? This is the structure and functions. It provides mechanical stability, right? As I said, in the cardiac muscle cells, due to contraction and relaxation. Okay, next, it facilitates cell to cell adhesion and signaling. So by these protein molecules, the cell to cell signaling can also be done, and the cell adhesion means joining, right? And important in tissue development and maintenance. It is very important in the tissue development and maintenance of the tissues. So next one, desmosomes. Okay, what are desmosomes? Desmosomes is a type of cell junction. Here also the cadherins and catenins are present, but the cadherins same. The cadherins of one cell is attached to the cadherins of another cell. The cadherins are present in the plasma membrane of each cell, and these cadherins attached to one another, and they are attached to adjacent cell. But inside this, the cadherin molecules, they are attached to the keratin molecules. Okay, the keratin intermediate filaments which are the keratin, they are attached to keratin, and it provides the structural support inside the cell. By this mainly where we can found in heavy quantity means in this you can see in this skin and heart tissue, where prevents cell from being pulled apart, especially in tissue subject to mechanical stress, like imagine this is a blade and this is your skin, right? This is your skin. If I placed a blade and rubbed on your skin, it will not harm to you. Why? Because the skin will be the skin is having some elasticity. It will move along with the blade and ensure that you doesn't have a cut, right? Why? Because of the presence of desmosomes junction in your skin tissues, skin cells. Therefore the skin has some elasticity, and it also prevents cells from being pulled apart, right? This is the main reason. And it also means the tissue integrity, right? Tissue integrity means it prevents the cell from being pulled apart. It is mainly present in the myocardium and epithelial tissues, right? As I said also already. So moving to next is the gap junction. Now gap junction is a special junction. If I give you a very common example, then you understand it very easily. You have already studied what is the synapse, right? You have already studied the synapse. Synapse is a space between the two junctions, where axon and dendrite of the two neurons will meet, and thereby the passage of signals or electrical signals will take place, right? It is the space between where two axons, one axon and dendrite meets, right? So have you ever wondered why we are saying synapse is a space? Because, for example, it is a axon of a cell and it is the dendrite of a cell, right? It is the dendrite of a cell. Okay, this is a neuron. A rough example I am giving you. So this axon is not completely attached to the cell of the wall of the dendrite. Why? Because it is a gap junction. It is a classical example of gap junction, that is the synapse, right? So how does it takes place? Basically, there will be a intracellular space of usually 2 to 4 nanometers. Between these, the size of this space will be usually 2 to 4 nanometers, right? Now in this 2 to 4 nanometers, there are some, in the plasma membrane there are some connexin proteins present. Okay, connexin proteins will be present, and there will be some hydrophilic channels. When the, like you can see, it is a closed and this is the open. When some molecule has to be passed from the plasma membrane, from the cell, then this is passed over here, and this connexin protein will be opened, channel will be opened, and when this opens, the molecule will pass from here, and due to the narrow space of 2 to 4 nanometer, it will just fly over to the connexon of the another cell, and hence the passage of a molecule of or any substance from one place, from one cell to another takes place, and this is a very rapid process, right? So here it is present that it allows direct communication and passage of small molecules between cells, and it coordinates activities in group of cells, and it is important in cardiac muscle. So basically take this example only. If it is supposed to be a heart, right? If it is supposed to be a heart, then for the contraction and relaxation of the heart muscles, the electrical impulses has to travel from all over the heart, right? All over the cardiac muscles, the electrical impulses should be traveled so that the contraction and relaxation can be occurred. So if when the electrical impulse is generated, then it has to travel from one cell to another cell, then another rapidly. So when it has to transfer rapidly, when the cell membranes are present, and from one membrane to another, it cannot pass through by active diffusion or passive diffusion, have you studied, right? So how they will be happening? Because they will be happening because of the gap junctions. From the gap junctions, the electrical impulses can be easily passed from one cell to another, and also the rhythmicity of the heart will be maintained, right? Why the rhythmicity? Because uniformly the contraction and relaxation has to be in a coordinates, right? In a rhythm it should occur. Therefore, by the help of the gap junctions, the electrical impulses can pass very fastly and the rhythmicity of the heart can be maintained. So the last one is hemidesmosomes. As I have said, it is a cell matrix junction. No, what the cell matrix junction? As the name suggests, it is a junction between the cell and the basal membrane, that is matrix. So integrin protein connect the cell to the extracellular matrix and intermediate filaments anchor inside the cell. What are its functions? I will show the diagram in the next slide. Functions: the anchor cells to the basement membrane and provides structural stability. You can see here. Hemidesmosomes are present here. So basically in a tissue, when a group of cells are attached, so basically this will be the structure. These are the hemidesmosomes. They are also interconnected, and the protein molecules are projected outside of the cell, which are attached to the basal membrane, right? This is the basal membrane, and attached to this basal membrane there are protein molecules which are projected outward of the cell, and they are arising from these hemidesmosome junctions, right? Now these are desmosomes because they are forming the complete structure, right? Here and here it is forming the complete structure. Basically hemi means half, so here half circle like junction is present, therefore it is called as hemidesmosomes, and here the circle is completely forming, therefore it is called as desmosomes. Now desmosomes is the junction tight cell junction present between adjacent two cells, right? And here is the adhesion belt, or you can say adherence junction, or it is also called as zonula adherence, right? Zonula adherence also you can say to the adherence junction, right? So this is about the cell junctions. In the next topic we can cover the next. Up to then, thank you. Keep watching our videos. Thank you.